Good morning, everyone. This is Mike Bellum. I'm the president of Life Scientific, a company I founded in 1992 representing manufacturers of parenteral production machinery. I wanted to take this opportunity to thank you all very much for attending this segment of the Life Scientific Lunch and Learn webinar series that will be presented today by Sergio Mauri of Fedegari. I do apologize for any confusion that may have occurred relative to the time of this lunch and learn. Perhaps they eat lunch a little earlier over in Europe than we do here. And, I guess occasionally we may have to rename it to the brunch and learn. But at any rate, I appreciate the opportunity. And for those that may not have been able to make the live scheduled event, the only sacrifice in that is that you might not be able to add your questions onto the chat segment here, but you can view this at any future point in time, and the links can be shared with any associates that might find the content interesting. So we plan on providing these lunch and learn sessions at least once a month, typically at 11:45 on the third Thursday of the month. And if you're interested in finding out what we're going to be presenting in our next several programs, just sign on to our LinkedIn page or the YouTube page, and you'll be kept current with the future lunch and learns that we're going to be doing. Just to get a little housekeeping out of the way, on the YouTube stream, you should be seeing a screen that looks similar to what is showing here, which is the live feed from our YouTube channel. So the stream is going through, and you have the option of selecting the full screen or going to the chat section. So if you go to full screen, you just click here. If you want to put a question up for Sergio during the content of the presentation, then you just will hit the escape button, go from full screen to the chat screen, enter your question here, and hit the send button. At the end of the presentation, all of the questions are going to be given to Sergio, and he will address what he can at the time. And then afterwards, offline, if there are any open items that we need to address, we certainly will. One thing that is unfortunate is that with XP machines, if you're not seeing the content of my presentation, it's due to a compatibility issue and browsers that no longer support XP. But the good news is that later, after the live stream has completely downloaded to our YouTube channel within probably an hour of completion of this, you'll be able to go back and review the content at any point in time that you would like. Certainly, if you feel any associates would benefit, this is quite shareable and will remain resident on our YouTube channel. So as I mentioned before, Life Scientific specializes in the representation of pharmaceutical production equipment, largely for parenteral applications. We're based in St. Louis and have offices and project engineers in Illinois and Minnesota. And our product line consists of everything from aseptic environments through container integrity testing. And over the years, our company's been involved in more than 5,000 equipment projects throughout the Midwest. So if you want any more detailed information on the companies we represent, get a hold of us. There are plenty of ways to do that these days. We began representation of Fedegari, several years ago now, they're a company that was founded in 1953, and they're focused on the production of equipment and technologies for pharmaceutical grade sterilization, washing, and decontamination systems. They've got extensive international infrastructure, including now 2 offices in the United States. Our presenter today is Sergio Mauri. He began working with Fedegari Technologies in 2010. Prior to that, in the early 80s, Sergio got his master's degree in chemical engineering, and he's been involved in aseptic environments and technologies ever since that time. Early in his career, he was involved in clean room HVAC design and systems engineering, becoming an engineering manager in that area. Then in 1995, he moved on to found his own company called Biocom, which was focused on the design, development, and production of pharmaceutical aseptic grade clean rooms and related technologies, including the development of a line of chemical decontamination systems, which he's going to talk about today. In 2010, his company, Biocom, was acquired by Fedegari Technologies, where Sergio is now responsible for aseptic manufacturing technologies. In the past and current, he's been a board member of the Association of Contamination Control, serving as its past president and vice president, and he's been real involved with the Parenteral Drug Association, as well as with regulators from the FDA and EU. Some of the papers and publications and courses that he's done over the years include the ASCCA training course on the general introduction of clean room technology, isolation concepts, and critical parameters definition. He's also been involved in training at a university level on HVAC design and engineering. He's done numerous presentations for the PDA. One of his recent presentations that was also published by Pharmaceutical Online was in gloveless robotic technologies for aseptic manufacturing of personalized cytotoxic drugs. For Fedegari, Sergio's been continuously developing surface bio-decontamination technologies, and today he's going to discuss the justification for the implementation of chemical decontamination in today's aseptic production environments. So with that, I'll turn it over to Sergio and thank him in advance for his time and coordination with us to hopefully help provide you some good educational content. Thanks so much, Sergio.
Thank you, Mike. Thank you, Mike, for your very kind introduction. So this morning in Italy, we are going to talk of a quite controversial issue, that is the transfer of material from the non-aseptic side to the aseptic side of a sterile environment. So the presentation outline, we are going to talk about different routing for this kind of transfer. And then we also emphasize that this kind of transfer can be quite a critical process. So we need to apply all the risk assessment approach in order to understand what is the best technique that we can apply to mitigate the risk. And then afterwards, we are going to go through all the surface biodecontamination process and the equipment key features, and also we, as final, having a picture of the system validation. Okay, let's move on. I would say that first initial talks, you know, what makes the difference in our world is even the small detail, because we need to take into account that everything that we are doing might be crucial for the health of the patient. We, as a paramount, we have to have in mind patient safety first. And this can be achieved by a good process and full compliance with the good manufacturing practices. So the routing of material transfer, there are several routing. Fedegari is very well known for steam sterilization routing of the material from a non-sterile to the sterile to the aseptic core. Also with dry heat sterilization for depyrogenation of glassware. Still, there are several transfer hatch with double door interlock. Radiate air, HEPA filtered air, and the transfer system, in this case I call, this is much more linked to the operation of the operator. So this is the reason why I call them spray and pray. Because you are wiping all the goods and the operator, it doesn't create a very robust process. And then in the last years, it was introduced the hydrogen peroxide vapor decontamination, and today we are going to talk of this type of routing. So, as I told you before, the material transfer issue is underlying the main guidelines from FDA, from EU Annex 1, and also for WHO. So we need to minimize the sterile area to the sterile area. And this, even if there is not mentioned the transfer hatch system, decontamination system, there are several in all of these documents are pointed out that there is a risk. So this we have to take in mind also the quality by design, and the quality by design tell to us that the quality should be built into the product itself, but also understanding all the process by which it is developed and manufactured. So we have to have a complete knowledge of all the process and the related features. So therefore, the material transfer into a aseptic environment takes an extremely important role in the quality by design approach. Also, we are supported by the ICH guide, the Q9 in this case, so quality system harmonization, that they are claiming for risk management. So we need to have an increased knowledge of exposure to risk of everything that can be exposed to a possible risk of contamination, foster quality by design, continuous improvement, and new technology introduction, which generally leads to enhanced product quality. So we have to be open to new technologies. This slide, just to give you a visual idea of how is complicated the aseptic process. So there are hundreds of elements that has to be taken into consideration in order to keep the our process under control. One of these elements is the material transfer in and out to an aseptic suite. There are some other drivers, by the way. The drivers that we got now are because there is a lot of outsourcing. So the company, the pharmaceutical company, trying to move some part of the process to other manufacturer, especially for the single use and the ready-to-use material. That this because using this kind of technology, they have less cleaning and downtime, so they are can be much more efficient. And also some other parts like stoppers or containers or clean room garments cannot be sometimes cannot be thermally sterilized. So all the material that are the only robust process to introduce into an aseptic suite is via an hydrogen peroxide vapor surface decontamination. Nowadays, the available technologies that I've seen around the world during all my traveling, I've seen nothing plus interlocked doors. Then I've seen spray and wipe and pray, as I told you before. Then we had some the more robust and validable system are with hydrogen peroxide vapor, with dry mist. With the dry mist, they're using peroxyacetic acid, chlorine dioxide acid, and nitrogen dioxide gas. To iodide because of the residual. So with hydrogen peroxide, after the decontamination process, we leave our environment with oxygen and water that are environmental friendly, also for our process. Here some load pattern example. In a septic suite, there is a huge amount of environmental monitoring plates. Swabs huge amount of this stuff in order to keep under control the process. In this case, we have aluminum kegs holding antibiotics sterile powder. So these kegs are directly connected to a dosing machine, and therefore I need to be sure that the external surface is not contaminated, doesn't bring any contamination from the non-sterile area. Tools for maintenance and also some other shipping and decontamination. So we need why we need a risk assessment approach? Because if we have this approach, there are some possible risks. So first risk, as I told in the first two slides, health damage by supplying a contaminated product, and this could be a real disaster. And then batch product rejection, and sometimes this batch costs a huge amount of money, millions of dollars. So losing a batch could be really harsh. If we lose a batch, then we can have a product shortage. If we have a contamination, then we need to make a lot of investigation in order to understand what is the source of this contamination. This means that my system will have a lot of downtime, and then inspection failure. So these are all the risks not having a good system. This just slide give you an idea of making a different risk assessment from spray and pray and UV plus unidirectional flow and hydrogen peroxide vapor transfer etch. On the internet, you can find some ranking table for different kind of risk. And then you can make a quick calculation. And as you can understand, applying a validated process like hydrogen peroxide, we really reduce the risk. We have a really high risk mitigation. This is the system can provide the process lethality, and the target of this system is to provide at least a 6 log reduction. Consider that the sterilization with the terminal autoclave can provide you a 12 log. Bio-decontamination material lock, first of all, the tightness of the chamber that has to be proved by vapor of hydrogen peroxide that can be dangerous or hazard for the then we need to have a good distribution of the hydrogen peroxide within the chamber. We need to monitor the concentration of the biocide in order to be sure that we have reached a certain amount of hydrogen peroxide. Also, we have chamber aeration, and this will happen through an HEPA filter. And to guarantee a chamber positive differential pressure during the standard or idle phases. We need to monitor the biocide effective removal that must be below the threshold limit value requirements, and everything has to be handled through a very robust control system. So the most used biocide, as I told you, are hydrogen peroxide vapor, peroxyacetic acid dry mist, chlorine dioxide, and nitrogen dioxide gas. So this is how it looks like the decontamination cycle. So the chamber of the transfer etch is first conditioned. So it means that we have to start with a certain amount of relative humidity. And for this reason, we add some clean compressed air with a dew point of minus 20 degrees Celsius in order to be sure that there is no water inside or just a limited amount of water. Then we add the biocide until we reach the holding time concentration level. Then we have the holding time that I wanted to achieve, and then afterwards we will have the aeration phase, in which we are going to inside the chamber until we reach a level of less than 1 ppm. And here we have a sketch of the system. So we have the compressed air, an air heater to heat up the compressed air after expansion. So this compressed air goes into the HP vaporizer. Where delivered to the enclosure. This is an enclosure with a unidirectional airflow. There are two different possibilities, one with a turbulent flow and the other with a unidirectional airflow. Very similar what's happening into the isolator. Then after the hydrogen peroxide passes vent into the exhaust in order to build up not any pressure. And we have an HEPA filter in case of electric breakdown. We can protect the content of the enclosure from possible contamination. When the aeration, we need to start with the aeration. We close this valve from the vaporizer, then we open the fresh air valve, and we will have a single pass airflow system. So all the with an exchange rate of about 200 chamber volume per hour, and then exhaust through a catalyzer, and then to the atmosphere. So some picture of this kind of equipment. You can have a small chamber, or even some pass-through chamber, floor standing. So you can introduce even trolleys inside the transfer hatch. Here we have the vaporizer. We have the dosing pump, hydrogen peroxide. This is the buffer tank that holds a fixed amount of hydrogen peroxide in order to be sure that we can finish the cycle that has been launched. Important point. It is important to point out we don't need. We are not selling hydrogen peroxide. This you can buy this hydrogen peroxide from a supplier. It's important that it has to be pharmaceutical grade, but there is no any link with us. The other important point is that the concentration inside the chamber is driven by a feedback loop. So we will have one sensor, process sensor, that it is capable to detect the concentration inside according to this sensor. The controller can change the number of strokes of this microdosing pump, and then adjust the amount of hydrogen peroxide that is delivered to the chamber. Important to notice is that even if the hydrogen peroxide is not really fresh, and it's a little bit degraded because of our control loop, feedback loop control system, we don't care, because we inject the amount of hydrogen peroxide until we reach the target of concentration within the chamber. So these are the biocide probe. We have one high concentration dragger, and then this high concentration switch to the low concentration during the aeration, when the aeration phase has reached a concentration that is below around 200 ppm. So the last part of the aeration is controlled by the low concentration sensor, from 200 ppm to less than 1 ppm. As we have designed a new door for this transfer hatch. This door has a dual inflatable gasket to be sure that no leaks can occur from the door. And in between the two gaskets on the door frame, more or less in this position, there is a small hole that inject in between the two inflated gasket, clean compressed air. So we are going to make a kind of bubble, help us to control the efficiency of the gasket, even if there is some debris on the doorstep and the gasket doesn't then there is a leak of compressed air, and this is detected by a flow meter, and we get an alarm of not proper seal of the door. So the aeration, as I told you before, is made with a single pass air removal with fresh air. Generally, it is size in between 150 and 300 air changes per hour. Considering that one of these transfer hatch is 1 meter cube, it means that the total flow rate is roughly 300 meter cubes per hour. In order to reduce the relative humidity and allow a ramp up of the concentration very quickly. And also we have in our system, we can rise a little bit the temperature, and the temperature gives some energy to the surface that has absorbed the hydrogen peroxide, especially the plastic surface, and this energy overcome the adhesion of the hydrogen peroxide molecules, and these molecules are stripped out. In case we want to reach a very low level of in the range of ppb, when we reach around 2 ppm, then we have to increase the relative humidity in order to take advantage of the polarity of the water molecule and the hydrogen peroxide molecule, and this polarity help to strip out from all the of the porous of the material, the residual H2O2, and with this system we can go down at the ppb level. Okay, here it's important to mention that Tyvek could be a problem with hydrogen peroxide. Tyvek is a porous material. You know, you use Tyvek bags also to make sterilization of and it is permeable as well with the hydrogen peroxide vapor, and this membrane keep quite a high quantity of hydrogen peroxide into the container in which the Tyvek is the lid. So the aeration is almost impossible with the Tyvek lid, and this has to be taken into account contamination of ready-to-use vials or syringes, because the residual of maybe several ppm inside the can provide big issues with the product, because they are oxidizing the products. So we arrive almost at the end. How can I make the validation of this system? In an evaluation of the enclosure with the load pattern study. So we need for instance, I cannot sit a container on a hard surface, because below on the lower part of the container, the hydrogen peroxide cannot enter, cannot penetrate. So I could have issues in making the decontamination of this part. All the shelf are wired shelf in order to minimize the contact with the product that are supported, and to guarantee the better distribution of the hydrogen peroxide. Also, we make air distribution studies with the smoke in order to check this load pattern. We need to validate the enclosure pressure decay test. So we need to be sure that the enclosure is airtight. Leak can occur. Making temperature relative mapping of the enclosure. Of course, as it's a HEPA filter leak test, and also particle counting, because we can guarantee an ISO 5 at rest condition inside the chamber. Afterwards, we have to move to the biocide uniformity evaluation. Therefore, we are making temperature mapping with the biocide with the vapor inside. This to check if there are some kind of stratification. The steam doesn't mix very well with the air that is inside, so it tends to go in the upper part. So the upper part might have a higher concentration than the lower part, and this can be with the chemical indicators that are in order to identify the worst location. After biocide uniformity, chemical, I would say, biocide uniformity, then we move to biological challenge. So we are working with the 3 BIs per location. This is because we can make a statistic approach of the D value location by location with the 3 BI. And this helps us also to make the calculation of the enclosure of the system of our transfer hatch D value. So D value change from one unit to the other unit, and we have to make the calculation for each single unit, and then the BI also can show us what is the worst location. At the end, after we have identified the worst location, we make the performance qualification with the BIs exposed for 3 times in the worst location. And the system, if we succeed, the system is biologically validated. In the meantime, also we are making study for the aeration, and also making biocide threshold limit value analysis. Okay, sorry, I went quite fast, but I want to leave some times for a quick answer for the Q&A time. The conclusion is that in pharmaceutical sterile manufacturing, disposable materials, heat-sensitive disposable materials, are becoming widely used in this process. The material transfer has to be designed to mitigate. We need to apply quality by design and risk assessment approach. Traditional material airlock cannot guarantee a safe and valid suite, and cannot be accepted anymore. Surface chemical biodecontamination is now the front edge of the transfer technology. By the way, just a few months ago, we had 2 times FDA inspector in our facility in Pennsylvania, and we provide a training on this technology, and they were from this kind of technology. Thank you so much. Mike, it's
Thank you very much, Sergio. We really appreciate it. We have a couple of good questions, and perhaps you can see those if you care to address. Sergio, are you able to see those, or would you like me to read them for you?
Let me check. We mainly debate on biodecontamination with hydrogen peroxide, which is very complicated. That would you recommend as more effective treatment against the microorganism? But you know, it's. I would say I could confirm it's that this hydrogen peroxide decontamination is. I would say it's tricky, not complicated, because you have to take care of several parameters, and you have to be sure that all these parameters are under control during the process. You know, it can just the change of 1 degree Celsius plus or minus you can get a different result. But this is especially if you don't have a kind of feedback control from this is provided by our system. So even if there is a change in the temperature condition inside the chamber, we have to reach a target of hydrogen peroxide level. So maybe it's a question of more minutes. The cycle will last more minutes. At the end, we are capable to reach the target. Okay. Do you think that steam sterilization autoclave is one of the most effective methods for decontamination biohazard material? Or there is a newer, more advanced solution? Okay, I think you know the hydrogen peroxide decontamination is just a surface process. If you have your material contaminated and this contamination is held in hollow parts of for the hydrogen peroxide to penetrate inside, or I would say almost impossible. So this is the reason that most of the time is used the heat for making the decontamination of biological matter.
If there are any additional questions that you or associates think of later, please go ahead and post them and we can address them at a later point in time. And I'd like to very much thank Sergio and Fedegari for their time and involvement in putting on this lunch and learn or brunch and learn for us. As previously mentioned, Fedegari has good domestic infrastructure. Sergio had mentioned the FDA in the facility. This is what we have in Sellersville, a complete showroom with several pieces of process equipment that are fully installed. This is a great opportunity for clients to come in, see the equipment, which can even sometimes in your own plant be very difficult to do. So we have this 8 to 5, come on, take a look at any of the equipment. We also have moved technology from or expanded technology from our training center in Pavia, Italy, into the United States and can perform cycle development services out of the U.S. office. So we've got a great showroom, a great training center. If there's some kind of course that you would like to have specially designed for your facility, we can certainly accommodate that. And just let us know if there are any training, validation, technical service training topics that are of interest to you, and we'll be happy to try to accommodate. Our next virtual lunch and learn meeting is going to be held August 16th at 11:45 Central Time, and it will be presented by Bonfiglioli Engineering on headspace gas analysis that has direct application for lyophilization technology, looking at the reduced humidity and oxygen levels that are typical in a lyo vial, but any reduced oxygen vial that needs to be analyzed, we can do that at 100% up to 1,000 parts per minute. So Bonfiglioli is going to discuss that technology and the applications of that technology. So please don't hesitate to sign up for that. We hope you found today's program beneficial. And if you need to address, like I said, any additional questions to us or Fedegari, please don't hesitate. And again, my thanks to Sergio for taking his time here today. We hope you found it to be quite beneficial. So thanks again. Take care.
Thank you. Thank you so much to all. Thank you. And hope talk to you soon. Present to you soon.